So the chapter 6, that is anatomy of flowering plants, the tissue system, anatomy of dicot and monocot plants. So study the internal structure. Uh, the internal structure and the functional organization of higher plants. So this chapter introduces you find to the internal structure and functional organization of higher plants. The study of internal structure of plants is called anatomy. So different organs in a plant show differences in their internal structures, right? The different organs in a plant show differences in their internal structures. Mm. So within angiosperms, the monocot and dicots are also seen to be anatomically different, and internal structures also show adaptations to diverse environments. Different. So monocot and dicot can be different, right? Anatomically different. So the tissue system. Uh, so types of tissues based on the types of cells present. Tissues vary greatly on their locations in the plant body. Their structure and function would also be depend on location. So their structure and function would also be depend on location. So on the basis of their structure and location, three types of tissue systems: epidermal tissue system, ground or fundamental tissue system, conducting or vascular tissue system. Now epidermal tissue system, uh, the like comprises epidermal cells, stomata, the epidermal appendages, that is trichomes and hairs. Now epidermis, it forms the primary plant body made up of elongate complexly arranged cells which form a continuous layer. Now it's single layered with parenchymal cells with a small amount of cytoplasm lining the cell wall and a large vacuole, right? Now there is cuticle outside of it, which is waxy thick layer, and this prevents the loss of water. So cuticle is absent in roots. Now stomata. So stomata, it regulates the process of transpiration and gaseous exchange. Now each stoma is composed of two bean-shaped cells known as guard cells. Encloses the stomatal pore. Now in grasses, these are dumbbell shaped, and the outer walls of guard cells are thin, and the inner inner ones are thick, right? And then these guard cells possess chloroplast, okay, and regulate the opening and closing of stomata. Now sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known subsidiary cells. Now the stomatal aperture, and the guard cells and the surrounding subsidiary, okay, stomatal aperture, that is stomatal pore, then guard cells and the second and the surrounding subsidiary cells together form the stomatal apparatus, right? Now you see epidermal cells, then subsidiary cells, then guard cells, then chloroplast it has, and then stomatal pore. So stomata with bean shaped guard cells in the first diagram, and second one is for the grasses, stomata with dumbbell shaped guard cells. Now the cells of epidermis bear a number of hairs. Root hairs are unicellular elongation of the epidermal cells, okay. Now on the stem, epidermal cells are trichomes. Now trichomes in the shoot system usually multicellular, branch or unbranched, and may be stiff or soft, may secretory in function. Now they help in preventing water loss due to transpiration. Now the ground tissue system. So all tissues except epidermis and vascular, that is conducting tissue systems, constitute the ground tissue, right? Now it consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Now parenchyma cells present in cortex, pericycle, pith, and medullary rays in the primary stems and roots. In leaves, the ground tissue systems consist of thin wall chloroplast containing cells and is called the mesophyll. Okay. Now the vascular tissue system. The vascular tissue system consists of a complex tissue, that is phloem and xylem. Now in dicot stem, cambium is present between phloem and xylem. So because this cambium is present, now they are the possess the ability to form secondary xylem and phloem tissues, and hence they are open vascular bundles. Okay. Now in monocot, vascular bundles have no cambium present in them since they do not form secondary tissues, so they are referred to as closed, right? So uh, when xylem and phloem within a vascular bundle are arranged in an alternate manner along a different radii, so these are the radial. So these are like radial. So these are like radial vascular bundles. Okay. So these are present in roots, right? Then conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves, that is conjoint vascular bundles, stem and leaves along the same radius. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. Okay. So various types of vascular bundles, as you see, first in the radial, that is xylem and phloem, they are at the same radius. Then they are at the alternate radius. Then second is the conjoint closed. It is for like monocot. Like in dicot, there is cambium between phloem and xylem, open for secondary growth, right? This is open vascular bundles. So now anatomy of dicot and monocot plants. So dicot root, we study the sunflower root, right? Now many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of cortex is called the endodermis. Now, this comprises a single layer of barrel-shaped cells without any intercellular spaces. Now, the tangential as well as radial walls of endodermal cells have a deposition of water impermeable waxy material suberin in the form of Casparian strips. Okay. Now, next to the endodermis lies a few layers of thick walled parenchymatous cells referred to as pericycle. So, next to the endodermis lies a few layers of thick walled parenchymatous cells referred to as pericycle. Now, initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. Now, the pith is small and inconspicuous. Then the parenchymatous cells which lie between the xylem and phloem are called, these are the conjunctive tissues, right? Which lie between the xylem and phloem, these are conjunctive tissues. Now, there are usually 2 to 4. There are usually 2 to 4 xylem and phloem patches. So there are usually 2 to 4 xylem and phloem patches. So later a cambium ring develops between the xylem and phloem. So all this tissue on the inner side of the endodermis, such as pericycle, vascular bundles and pith, constitute the cell. So vascular bundles, pericycle and pith. Pericycle, vascular bundles and pith constitute the stele. Now monocot root. So the anatomy is almost similar in many aspects. Okay. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6, that is polyarch xylem bundles in the monocot root. Now pith is large and well developed, and monocotyledonous roots do not undergo any secondary growth. So this is monocotyledonous root. So first one is dicot root, that is primary, then monocot root, right? Root hair, then epidermis, cortex, endodermis, pericycle, protoxylem, metaxylem, pith and phloem. Root hair, epidermis, cortex, endodermis, pericycle, phloem, protoxylem, pith and then metaxylem. So this is the dicot. Now we are going to study the dicot stem. So dicot stem, okay. So same the epidermis, okay. Now covered with a thin layer of cuticle, okay. and it may bear trichomes and a few stomata. Okay, now the cells arrange in multiple layers between the epidermis and pericycle. So between the epidermis and pericycle constitute the cortex. Now it has three subzones. The outer, that is the hypodermis, constitutes of few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Now cortical layer below hypodermis consists of rounded thin-walled parenchymatous cells with conspicuous intercellular spaces, right? With conspicuous intercellular spaces. So it is called the, it is what? It is cortical layer. Okay, so first one is the like hypodermis, then cortical layer, and then below it is there is cortical layer below hypodermis constitute an innermost layer of cortex, that is endodermis. Then third layer is the endodermis. Innermost layer of the cortex in dicostem is the endodermis. Now the cell of the endodermis are rich in starch grains, so this is why this layer is known as the starch sheath. Now pericycle present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles there are few layers of radially placed parenchymatous cells which constitute medullary rays. In between the vascular bundles there are few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of dicostem. Now each vascular bundle is conjoined open and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. So the pith is large intercellular spaces which occupy the central portion of the stem constitute the pith. So pith is very large, right, in here. Now monocot stem. In monocot stem it's written that like the hypodermis is sclerenchymatous and large number of scatter vascular bundles. Now these scatter vascular bundles surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Right. Now the vascular bundles are conjoined and closed. Now the peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water containing cavities are present within the vascular bundles. So this is the monocot stem, right? Now the dorsiventral, that is dicotyledonous leaf. Okay, so there are like three mainly parts, that is epidermis, mesophyll and vascular system, that is xylem and phloem. So epidermis, which covers both the upper surface, that is adaxial epidermis and lower surface, that is abaxial epidermis of the leaf. They both has a conspicuous cuticle. Now abaxial epidermis generally bears more stomata than the adaxial, that is upper one. Its opposite, lower has the more stomata. The latter may even lack stomata. Now the tissue between the upper and the lower epidermis is called the mesophyll. Now the mesophyll, which possess chloroplast and carry out photosynthesis, is made up of parenchyma. So they are made up of parenchyma. Now it has two types of cells, the palisade parenchyma and spongy parenchyma. Now the adaxially placed palisade parenchyma is made up of elongated cell. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extend to the lower epidermis. There are numerous large spaces and air cavities between these cells. So now the vascular system includes vascular bundles. The size of the vascular bundles are depend on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. Now the vascular bundles are surrounded by a layer of thick walled bundle sheath cells, right? Now the isobilateral, that is monocotyledonous leaf. Okay, the anatomy of isobilateral leaf is similar to that of dorsiventral leaf in many ways. Okay, the stomata are present on both the surfaces. Now the mesophyll is not differentiated in palisade and spongy parenchyma. Now in grasses, certain adaxial, that is on the upper side, epidermal cells along the veins modify themselves into large empty colorless cells. Okay, these are called the bulliform cells. Now when the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed, and when they are flaccid due to rest, they curl inward to minimize the water loss. Now the parallel venation in monocot leaves is reflected in near similar sizes of vascular bundles, so except in main veins as seen in the vertical section of the leaves. So this is it. Okay, the ground tissue forms the main bulk of the plant. Monocotyledonous and dicotyledonous plants show marked variation in their internal structures.